Welcome, everyone. It's very exciting to be here at the law school in a Helen meeting that's not completely virtual. It's good to see people's faces. I want to start today by acknowledging that we are meeting on unceded Aboriginal lands. I'd like to pay my respects to the traditional custodians of the land in which we're meeting, which are the Wurundjeri people of the Coghlan Nation, and we pay our respect to their elders past, present and emerging. Today, we have a very exciting Helen seminar to bring you on AI and automation in genomics. And it's my pleasure to introduce two of my colleagues and good friends, Danya and Simon. I guess I'll introduce you both now, and then I'll hand it over to you. So first, Danya is a Senior Research Fellow and Team Leader at the Biomedical Ethics Research Group at MCRI. She holds an honorary position at the University of Melbourne as a guest professor at the Centre for Biomedical Ethics and Law at KU Leuven in Belgium, where she first did her postdoctoral fellowship. She is the new co-chair of the Education, Ethics, Social Issues Committee of the Human Genetics Society for Australasia and an active member of the Global Alliance for Genomics and Health. And I would just say, a world leader in how genomics is sort of delivered to people and the ethical issues that raises. And we also have Simon. Simon is a moral philosopher and veterinarian. He is a senior lecturer in the Centre for AI and Digital Ethics and the School of Computing and Information Systems at the University of Melbourne. Simon's research in digital ethics currently includes projects on care robots for older people, animal-computer interactions, and digital technologies in healthcare and public health. He has an interest in applied ethics of various types, including environmental ethics and medical ethics. So with that, I will hand over to Danya.
So we're going to do some tag teaming today. I'm going to start off by telling you a little bit about what genomics is, and then we're going to launch into how AI and genomics might work. And then I'm going to present some data at the end of it about a study I've just done, which the title is up there. Genomic sequencing is actually a combination of a bunch of different genomic technologies. We call them next generation sequencing technologies, and it involves whole genome sequencing, which is where, as you might expect, you're sequencing most of the genome. Whole exome sequencing, which is where you're just trying to sequence the coding regions of the genome, so the bits that code for proteins. And then gene panels, which is where you're looking at very specific targeted groups of genes that might cluster around a particular phenotype. So it's increasingly being embedded in healthcare. It started off being identified or being used just mainly in gene identification in rare diseases, but it's moved into the cancer field. And now it's spread into a whole bunch of other subspecialties like cardiology, neurology, nephrology, etc. And that has a lot of implications in terms of who might be offering those tests, because it means that people who don't have genetics training might be offering testing to patients, and they might not have the skills in order to be able to explain the technology very easily. So there are a bunch of benefits of using genomic sequencing technologies over other standard sequencing technologies. And basically, it really does increase the rate and chance of gene discovery in research settings. So before we were doing sequencing where you're just sequencing one gene at a time, because you're sequencing many, many genes at the same time, it means that you have a much better chance of finding the gene that's responsible for a condition. So therefore, it increases the chance of diagnosis for patients, but it also then reduces the time to diagnosis because you don't have to go through and sequence gene by gene. And that can have a lot of implications in terms of being able to implement treatments a lot quicker. And it also reduces a lot of invasive investigations where you might have to do like a muscle biopsy or something to do a test in the past, and now we're using sequencing as a first-line test. We don't have to do that kind of stuff. But it does have some repercussions, and one of those is that you might find incidental findings. So that is where you have a change in the gene, a gene that is disease-causing but unrelated to the clinical question that you're asking of the patient. And so it might be something like you're doing a sequence for try to find an eye condition in a patient, but you find like a breast cancer gene or something. So that would be an incidental finding. But we also have the chance of finding what we call variants of uncertain significance. And those are changes in the DNA where we don't have enough evidence to say whether it is or is not causative. So it's sort of in limbo land, and that we have a lot of trouble knowing whether to tell patients about that because it's hard to understand what that might mean and the significance of that. But despite the fact that there are lots of benefits of using genomic sequencing, there are actually still a lot of undiagnosed patients, and that's where I'm going to launch into why AI might be helpful. So genomic data is the product of genomic sequencing, and that can be stored for a long time because it's not like a sample. It's actually DNA. So it's not DNA, it's actually data. And we know that test interpretation changes over time. So a lot of those variants of uncertain significance that I was telling you about, they might change status to either knowing that they're benign or pathogenic over time because we have new knowledge in the field. And we're trying to think about genomic data as a healthcare resource, which can be used both for the patient in front of you, but also if you share that data with other people in databases, then that can actually help diagnose other patients as well. And so reanalysis is something that we can think about as well. So we might want to reanalyze data down the track. So you might have a lot of undiagnosed patients to begin with. But then if you reanalyze the data over time, because we have new knowledge and we have new other patients we've got on databases, then we might actually be able to increase the chance of diagnosis. And this circle just shows that there are actually lots of different types of reanalysis, which I won't bother going into, but it's actually, as a term, it's a little bit complicated. So we have a lot of evidence that shows that if you actually do reanalyze the data over time, so the standard is about 22 months, you can actually increase the diagnosis by about 15%. And so that's quite a large increase when you think about how many patients that might be over time. So if we've got like thousands and thousands of patients that have sequencing done, and only about 50% of those might have a diagnosis, an increase of 15% is actually quite huge. And there are a bunch of different recommendations internationally that have said that reanalysis is really important, and they've actually recommended it should be done every 2 years. So as I said to you before, there's a lot of undiagnosed patients. So over the period of 2018 to 2021, there were 25,000 genomic tests that were done in Australia. And of those, 17,000 remain unsolved. So that's a lot of patients that don't have a diagnosis after having genomic sequencing done. And as I've just said to you, that if we know that if we analyze after about 2 years, we can get an extra 15% of those patients diagnosed. But the problem is that if we go at the current rate, it will take us actually about 85 years if we just go manually like we're doing it now, reanalyzing by hand, because it's actually really time consuming for laboratory scientists to do. So what we're proposing, and there's some work being done at the moment that I'm part of, which is to try and implement an automated reanalysis process, which would use AI technology. And that is the reason this is so huge is because there is just so much genomic data that comes out of every single sequence. So to be able to use AI in this process would actually really help. So I'm going to hand over to Simon now.
Thank you. Hi, everyone. Thanks for coming along, and hi to everyone online as well. So I'm going to do a little segment here on AI before handing back to Danya for her to finish off. So I work in the area of AI ethics and digital ethics over there in the School of Engineering. And let's click to the next slide, please. And just a brief introduction to artificial intelligence. Does anyone work in artificial intelligence or know a whole lot about it? Any experts in the room? No, good. Yes, yeah, just wanted to check. So AI is typically defined as digital technologies that can solve problems somewhat like intelligent beings by doing things like making inferences, classifying things, and making predictions. So modern AI techniques involve machine learning, or at least many of them do. And machine learning involves training an algorithm on usually a large set of data, sometimes a vast set of data, to find really complex and intricate patterns within that data and correlations which a human being might not be able to discover, or will never be able to discover because it would take too long and be too difficult. So as an example of a recent AI technology that performed complex tasks within medicine, there's the AlphaFold example, which used a deep learning technique to predict the folding structure of 600 million proteins just from their amino acid sequences. So it's a pretty impressive achievement. No human could ever do that within their lifetime. So AI is moving quickly into medicine, as well as a whole lot of other areas of life, of course. And it's been proven to be very effective at making certain predictions and diagnoses. So, for example, it's as good or better than humans at detecting retinal disease in diabetic patients. It's as good or better than humans at detecting breast cancer from pathology slides. And in some experiments have found that it's better than a number of radiologists at predicting whether a lesion on an X-ray is lung cancer or not. So it's making some pretty big strides there into medicine. So within pediatrics, which is what we're focusing on here, and I should just actually interrupt myself here to say that, which I forgot to do before, that Danya, Chris and I are working on this area, looking at pediatric genomics and how AI might raise ethical issues in that space. But in pediatrics, it's been shown that AI can be very good at identifying sepsis, pulmonary hypertension, things like autism, asthma and cancer. Within genomics itself, AI has been used to diagnose people in ICU, including children who are critically ill and where a diagnosis is urgently needed very quickly. AI can also be used, has been applied to predicting a genotype from a given phenotype. So, for example, to predict what genetic disease a child with certain facial deformities might have. And to do the converse as well, so to predict phenotypes from genotypes. So, for example, a fetus that has a certain rare mutation, AI can be used to predict what disease that fetus may go on to acquire. So in coming back to genetic or genomic reanalysis, many of these conditions that patients suffer from have unknown genetic causes. And some authors, like this one here, De La Vega et al., have said that ideally, all these unsolved cases would be reanalyzed automatically, periodically. And a subset with high likelihood of new findings would then be prioritised, handed over to humans to perform a manual review of those predictions. And this could be useful because it's a labour-intensive business and a costly business to do all this manually. So automation seems to pose benefits there. And AI could be used to do this on a continual basis, round the clock, as it were. So I want to now, just having given that little bit of an introduction to what AI might be used for, to raise some ethical questions to do with AI and genomics. Within the field of AI ethics, it's common for these kinds of principles to be mentioned. 4 of these principles are basically pinched from Beecham and Childress's classic 4 principles of autonomy, beneficence, non-maleficence and justice. And people, AI philosophers have introduced other principles as well into, working out whether AI should be applied in this or that way. And Floridian Cowles have introduced this principle called explicability. Explicability referring to the ability to understand what an AI prediction, the reasons for the AI's prediction, which we'll get onto in a minute and I'll explain. So let's start with beneficence and question of benefit. So the idea here is that AI should provide some sort of benefit, and certainly in medicine, using these technologies should be able to provide some kind of benefit. What kind of benefit could they provide? Well, one benefit of AI and automation would be that it can reduce human errors. So humans are prone to things like tiredness, distraction, and to a range of cognitive biases. So AI could take some of those things out of the picture and therefore result in better outcomes. It could also incorporate evolving evidence, perhaps more readily than humans can manage, from databases of genetic disease, and therefore lead to, one hopes, more accurate variant detection. And then subsequent to that, perhaps better diagnosis as well of genetic conditions. So benefit of this is that it replaces the labour-intensive and resource-intensive manual searching for these conditions, makes them quicker and reduces the cost of them, and can happen continuously. And a final benefit there that we will mention is the fact that some genetic conditions are actually rare and may be missed, therefore, by human operators, whereas an AI system may be able to pick those more rare conditions up. So that's benefits. What about the harms, though, of AI for these kind of purposes? Well, I mentioned a moment ago that AI can reduce bias, but humans are actually prone to a kind of bias involving artificial intelligence and automation, a kind of automation bias, where they're more likely to trust the deliverances of an AI system simply because it is artificially intelligent. And that could obviously cause harm. Another issue that we're all familiar with at the moment is leaking of private health information or hacking into private health information, releasing that into the community or the dark web. And that's always going to be a risk with artificial intelligence and the kind of data that's required to train those algorithms, which may be kept in a database. And perhaps a rather strange kind of problem here or potential harm is the possibility of adversarial attacks on AI-based algorithms. So a malicious actor could introduce data into a system to corrupt that system and to make it produce unreliable predictions and answers. So there's a nice little example here, I think, where in research they've tried this out. An AI system can be extremely good at identifying, say, a panda. But when a malicious actor gets involved and introduces some noise into that algorithm, the AI system can make really terrible mistakes. So in this case here, it's identified the panda correctly here, but after the adversarial attack, it's mistaken the panda for a gibbon. Obviously, that's a problem if that happened with medical data and medical predictions. Another issue is that that AI is not perfect, so it can produce false positive diagnoses and false negative diagnoses, and overdiagnoses as well. And the problem with that is not just that it happens, but that it could happen at a bigger scale if automation is rolled out across many hospitals or many different settings. It's also recognised that AI technologies might do very well in laboratory settings when they're being trained and tested, but that doesn't always equal good performance in the real world. So there's always a need to test the application of the AI systems in the real world and to conduct reliable studies, say prospective studies that show whether or not they're beneficial. And the final harm there, or potential harm, would be that AI systems can either be trained and then locked, or they can be left open and trained on data as it comes in, as we were mentioning before. But the risk of that is that it might make the algorithm less reliable or less accurate. So that's harm. There are also issues of justice and fairness that AI poses, well, recognised ones. So AI predictions can sometimes be biased against certain groups, people, certain demographics. And that can result from the way that the AI is trained, and it can also result in the way the AI is applied. So if it's applied to different target groups, it might end up being biased against certain groups. So an interesting example of that, much discussed and cited, is by Obermeyer et al., who found that an AI system had recommended less treatment for Black patients than for white patients for precisely the same sort of conditions. And the reason for that was because they discovered it had been trained on data that involved historical biases against Black people. So another example here is that of a potential bias is, say something like skin cancer diagnosis using AI that performs better on white skin than on Black skin. Another example would be using AI in facial analysis that's better at diagnosing Down syndrome in European people than in people of African descent. So this has the problem to both create inequities and to compound already existing inequities. So how does that relate to genomics? Well, 70% of the existing genome-wide association studies are based on populations that have European ancestry. So, this could result in biased predictions. So for some minority groups, variants may be falsely classified as rare or as disease-causing, so the AI could get it wrong. And final part of justice and fairness in AI systems is the fact that some people might have access to these systems, whereas other people from less fortunate places may not have access to them. Well, I mentioned before that AI scholars have introduced this notion of explicability and even included it as an ethical principle that we should be mindful of when we're applying AI into medicine. So explicability here refers to our ability to understand the grounds of a prediction, why an AI made a particular diagnosis, say, our understanding of how that came about. And one reason why this might be difficult is where there are companies which own these systems, which don't tell us how the machines actually work and make their predictions. There's also a deeper problem with this, where you can have systems that are just uninterpretable. So you could contrast, for example, a more simple kind of AI system involving a decision tree versus a deep learning system that has a vast neural net that simply cannot provide the reasons for its predictions. And these are often called black box systems. So we can't really ask this system, why did you say that I have disease X rather than disease Y? It can't tell you that. So some people might say, so what? The key thing they might say is that these machines are accurate in making their diagnoses rather than explicable. Well, one of the problems with that line of thought is that it makes it very difficult or impossible to detect whether or not, at least in many cases, the system is in error or is biased in its predictions. And you know, so you might have a very accurate system that is sometimes unreliable. So it may make sometimes very strange and spectacular errors, as we saw a moment ago with the panda, which was misidentified as a gibbon. A mistake that no human would ever make. So this means that some patients and some practitioners might prefer not to use black box systems. They might prefer to have to use systems that are slightly less accurate, but that can provide reasons for their outputs. And some AI scientists actually think that we should try and avoid these black box systems completely in medicine. And to finish with, before I hand back to Danya, is the final principle. There's autonomy and the issue of consent, gaining consent for using, in this case, artificial intelligence in genomic diagnosis. We considered whether, you know, how this might apply to children and taking samples from young children to make diagnoses and then doing that on an ongoing basis as new evidence comes in to try and find variants there for undiagnosed diseases. So what this might require is that as the child matures and gains autonomy, that they are offered the opportunity to be involved in those kind of discussions about whether AI should be used in their diagnoses. And it also raises the possibility that the parents' autonomy might be infringed if these things are done automatically without their ongoing consent, and could potentially cause them distress and harm if the child has, in the meantime, reached the age of 18, or in some unfortunate cases, the child has died, and they're given this information about the child. So I think this means it's back to Danya.
So I did flag earlier that there was some work being done in this area, and this is just I won't go into this slide in a lot of detail, but there was a $3 million awarded for the Genomics Health Futures Mission, which is part of the Australian government's Genomics, I can never remember, MRFF. Yeah, but MRFF stands for Medical Research Futures Fund. So many letters. So there was a grant that was awarded to Professor Zonitza Stark and also Daniel MacArthur, and this is to try and develop an automated reanalysis program. And so it's very complicated, all the things that they're doing, but you can see down the very bottom, there's a little bioethics word there, and that's where we fit into the picture, always at the end for some reason. But it's a very important component, and the work I'm going to present to you now is part of the work I've been doing as part of that grant. So we really wanted to know, so we said, okay, we're going to automate this reanalysis process, even though that Simon's just outlined there are a lot of hiccups that we need to kind of work through with the ethical issues around genomics and AI being used in genomics medicine. But this is something, as I outlined to you before, it's a really big problem. We have so many samples that we need to have reanalyzed to try and increase the rate of diagnosis, and we can't do it as humans by ourselves. So this needs to happen. And so we really wanted to say to the members of the workforce, what do you think about how this is going to work and fit with your existing workflows? And the colleagues I've just listed there are other people, and Fiona Lynch is in the audience here, are other people who worked on this project with me. So the aim of the study was to explore the views of laboratory scientists and clinicians about a range of different things, but we wanted to ask about their attitudes and preferences relating to the implementation of AI and automated systems in reanalysis projects. We wanted to talk to them about how it might fit with their existing workflows to sort of figure out like where those barriers might be and those pinch points, and try and help figure out how we can work through those. Wanted to gauge the concerns that they had and see if they related to the things that we thought they might be worried about, and also just to figure out some of the ethical challenges that they were struggling with. So we recruited via our own networks. So Zornitza Stark is head of the Acute Care Genomics program in Australia. And so we recruited a lot through her existing systems through that group. With Fiona's help, it was a lot of work, but we grouped and scheduled a lot of the focus groups, and we tried to group initially by profession. So we tried to have lab scientists together and clinicians together, and we managed to do that for one focus group of each. And then it was a bit of a mess because everyone is so busy. But we managed to then, it was quite good actually to then have a combination of different professions in each group. So the focus groups ran for 1.5 hours, and I facilitated those with Fiona's help. And the first thing we did was present the workflow that we were planning on using as part of the automated system. So I'm just going to talk you through that now. So as I've said to you, there are lots of unsolved patients. So we'd start with an unsolved patient, and then we need to think about what triggers that reanalysis step. So at the moment, there are multiple ways that can happen. At the moment, it's often a clinician who sees the patient again. So the patient's still undiagnosed. They come back for a repeat appointment 2, 3, 5 years down the track. And the clinician might then say, let's have another look at your data. And they would go to the laboratory and say, can we reanalyze the data? So that's one way that can happen. It might be that the patient comes back and says, I've got some new symptoms. And so the clinician would then say, okay, let's reanalyze the data that way. So that's another way that can happen. But what we're proposing is that this would potentially not need a triggering step, that it would either happen routinely. So it would happen like maybe every year or 2 years, that all of the data would get reanalyzed, or that it's actually a continuous process where it's constantly being looked at by the AI system. So we wanted to gauge from the participants what they thought about those two potential options. And then there's issues around consent. So at the moment, we've got all these patients who have consent to have analysis done for their diagnosis, but sometimes their consent form, depending on where it was done in Australia, because every state has their own consent form, which is very confusing. But it might depend on that might dictate whether they actually have given consent for reanalysis. So there might be just a line on the form that says your sample might be re-looked at in the future in light of new evidence or something like that. So it's very vague. So we wanted to talk to the participants about what they thought their consent form said and how well they thought that consent was covered in those for reanalysis. But obviously, none of those consent forms say that reanalysis is going to happen by an automated AI system. So I wanted to talk to them about whether they thought that was important or not. But then, of course, should we be talking to these patients again and saying, before we do the reanalysis, that's going to happen? Do they need to be notified? So there's some of the things that we wanted to figure out in that first initial step. But then the reanalysis would take place. And then we've got another set of questions around how we would get results to clinicians. So would we just send it to the clinician who had initially requested the test, or if they're not seeing the patient anymore, how do we navigate that? Would it go automatically into the medical record? It would need to, but then what does that mean in terms of like other professionals seeing it and maybe passing on information that the patient doesn't know about yet? So there was a whole step around that. And then, of course, the patient needs to be informed about those results. And so we need to figure out who's going to do that. So as just mentioned, it might go back to the clinician, but should it go to the clinician that doesn't see them anymore? How do we figure out who else might be involved in their care? What if it's still negative? We still want to know, should they be informed that there was reanalysis done, it's still negative? That's a lot of paperwork to be sending out. And then we also need to think about like funding issues. So how are we going to be paying for all this reanalysis? I mean, the reanalysis itself wouldn't take that much because it's just a computer doing it, but all the other follow-up steps would obviously cost a lot of money. So we presented this to them and we said, this is what we want you to talk about. And so we audio recorded those sessions and they were transcribed. And then myself and Emily King have been starting to analyze that data, not reanalyze. just analyze it. But we're still in the early phases of that. So I'm just going to be presenting some preliminary data today for you. But we did have 21 participants, which is reasonable, and that was across 6 different focus groups. So we had quite an even split between clinical people and laboratory people, which was great. You can see there that we covered a range of different professions, so clinical geneticists who were trainees and fully qualified, and genetic counsellors as well. And then we had quite a lot of clinical scientists, medical scientists, and a couple of genetic pathologists. We were fortunate to get people from pretty much every state and territory, which is great. And so you can see there's quite a split there. Most of them were from Victoria, and that's because we have a lot of stuff going on here. We have like a very large centre and a lot of people working in that space. But New South Wales and Queensland also have quite a few people working in this area. And then you can see we had a split across different levels of years of practice. So most people were relatively new, so less than 5 years in the field, but we had some very experienced participants as well. So it was good to get a mix there. So just in terms of like the preliminary results, and then I'm going to zoom in on one particular aspect. Participants were mostly supportive of implementing automated reanalysis processes, but they did have quite a few concerns, as you can imagine. And a lot of it was to do with workforce capacity. So they were both laboratory scientists and clinicians were really concerned about how they were going to manage this system because, at the moment they've just got their manual curation and they're very much in control of that. But if they're doing a reanalysis in the background and it's spitting out results here and there and everywhere, and they would have to be confirmed by a laboratory scientist before they would get relayed to the patient. And then clinicians would obviously have to be involved in translating that information and giving it back to the patient. So they would be really concerned about how that would work and how they'd manage to be able to sort of squeeze that into their existing very overloaded workflows. There were quite a few concerns around the fact that they wouldn't have updated information about the patient. So firstly, they wouldn't have they may not have their contact details, depending on how long after the reanalysis, how long after the initial analysis, the reanalysis was taking place. So we're just talking about a few years at the moment, but this is going to be going on for a long time. So what if they've had in 2018, they had a sample taken, and we're talking about in 10 years after that? How do we know that if they're alive or deceased? And that's obviously quite a problem when you're talking about very young children who are very sick. And so we want to make sure that we're not bringing up stuff from the past for parents who don't necessarily want to know that information anymore, but maybe they do. And so how do we know that? And then there were also concerns around the fact that if they've had a child who's been very sick or maybe even died, and we find a diagnosis for that child, and then they're pregnant with the next child, and how do we know if they actually want that information? And it could be an issue if they're like very far into the pregnancy. What do we do with that? Could that cause more distress? But also in terms of things like life insurance, what does that mean if they've potentially even had life insurance policy and then cancelled it, and then find out a result after that? So there's lots of things to be aware of, but there's also a concern around the lack of up-to-date phenotypic information. So I mentioned before the scenario where a child might come back because they've got a new symptom or something like that. So all of that information about the child's symptoms goes into the analysis and the reanalysis as well to try and figure out what the cause is. And it helps narrow down the clinical picture, the genomic information with the clinical picture. So if we don't have up-to-date information, that might mean that we're actually doing ourselves a disservice because we might be looking at the wrong symptoms for that patient. But I want to just talk a little bit about the consent issue, because obviously there's a lot of people who like the idea of consent in this room. So one of the things that I flagged before is, do we actually have consent to perform the reanalysis? And there was quite a few people that said they didn't think that their existing consent forms would cover that. So this is just, I'm going to present some quotes. So this person said, this was a genetic counsellor, said, so I don't think all of those cases would have been consented in enough detail to be aware that this was happening, or to not be surprised if they're contacted about a result. That was a real concern for them. And there was concern that that lack of consent might cause delays in terms of like getting results back to patients, but also in terms of the whole process. And it raises a bit of an urgency that, all the data is there now for the last 4 years, but we haven't been getting consent. So we need to have the good consent form in place, but I don't think we've been forward thinking enough for that at the moment. So that was a clinical geneticist. And then we talked a bit about where the patient should be able to opt out of the reanalysis process. So we're talking about it being a very automatic process, but should they have the opportunity to say, no, I don't want that to happen? So there was a couple of people who thought that should eventually be automatic, but for now, they should have an opt-out process. And that was really because we don't necessarily have everything sorted out. There's still going to be a lot of things that we need to figure out in terms of accuracy and the processes. So they were concerned that if people didn't have an opportunity to opt out, they might be problematic for them. So this clinical geneticist said, my view is that the initial consent for testing should be an automatic inclusion of reanalysis. It forms part of the test, you know. This is kind of what you're buying into, but I guess we're treading into sort of fairly new territory because it's an ongoing process. We've not really had that before. In any other research paradigm, you also include that ability to withdraw at any point in the process. So I think we have to have that ability. And because of this idea about opting out, a dynamic consent process was highly favoured. So a dynamic consent, for those who don't know, is where you have a process where patients at any time can sort of pull the plug or say they want to come back in and change their preferences, sometimes even about what kinds of results they want to receive. So as this clinical scientist said, I think dynamic consent is a good thing, a good development in this area, instead of just obtaining initial consent and then carrying that out for 10 years or however long we're going to keep reanalyzing the data. And there was some talk about whether patients should be alerted when reanalysis is going to occur. So that's something that could happen potentially through a dynamic consent platform where they could get alerts by email. It's very hard to read, sorry. So this was a pathologist who said, if you have the perfect system whereby they're pinged if a reanalysis is going to occur, then they go online to their dynamic consent and they go, well, actually, yeah, that's okay. And I want to know incidental findings, that is, now as well. So I'm going to tick those ones. Or the opposite, I don't want to know anything. I only want to know primarily related to this variant of uncertain significance in this gene only that was flagged in the first report. Now that's practically difficult, but that's the idea. If we're talking about patient rights, that's the ideal point to start from. And then we talked quite a lot about why it might be important to get reconsent for reanalysis. And so, yeah, some people might not want their data reanalyzed. And so I mentioned some of those reasons before, but I'm just going to highlight a couple of those in a bit more detail. So this genetic counsellor said, there are some people that have this aversion to things being continued to be looked at. And in reality, in my experience, has been pretty low, like low percentage, very rare really. People are like, yeah, why not? Just keep looking. And on the rare occasion where you do ring them, they might have a vague recollection that we talked through this, and they're usually very grateful. But I think the odd small number of people that might not want that and specifically opt not to get more information are often people who have this underlying anxiety about who's looking at my information, why they're still looking at it, and how long is it retained and all that kind of thing. And so I flagged before that some people might have concerns about insurance. So this genetic counsellor said, thinking about insurance implications and things like that as well, I guess our knowledge is growing about the spectrum of the disorders as well and how that might be linked to other things. And we might not have prepared them for that information. And some patients might have got insurance before testing and then cancelled it, thinking that we didn't find anything or not renewed it, and then we give them another result. And then, as I flagged before, families might be having another child and that might be problematic in terms of timings. So this genetic counsellor said, and pregnancy is a really difficult one because often they were testing hoping to get information they could use and didn't find anything. Maybe pregnant, maybe at a really late stage of pregnancy, and then get a result which they can't do anything about. The question is, do you want to know that at that point in time? And I think it's really hard for patients to say that they don't want information when they know it's available, even though that might be detrimental. So that's just a snapshot of some of the issues around the consent, but obviously there was loads of other stuff that we talked about. And I just wanted to sort of conclude for both of us. So as Simon talked to you about, there are lots of benefits of implementing AI in genomics. So it's obviously a lot faster. Hopefully it's going to be more efficient. Hopefully it will reduce human error and it can look at new things and find new patterns that we wouldn't be able to see. But there are also some potential risks around transparency, inaccuracies that are potentially going to come out and whether we can pick them up because we might not know that they're inaccurate, accountability issues and also inequity. And we've just talked about automated reanalysis as a key example, but there are lots of other areas in genomic medicine where this could be utilised. But in terms of this particular example, as we said, health professionals and lab scientists were mostly supportive, but there were concerns about workforce capacity. There were lots of issues about this lack of follow-up of patients and what that might mean for both the analysis and also being able to recontact them. And they had a lot of concerns about whether the consent process was sufficient at the moment. And also, if you think about how many people there are, I just said there are 17,000 people that have undiagnosed samples. Having to go back and get consent from all of those people would be a nightmare. And that's where I'm going to leave it because that's where I'm stuck. But thank you. And we're happy to take any questions. Thank you.